Hi grade 10, so this is my second attempt to make this document reader recording a little bit better. I've made it lighter. Okay, so this is a diagram which also summarizes the stages in mitosis. So it could be mitosis in a plant or in a root, in any of the places that you've been learning about. Okay, so if we look at the diagram, we're going to start on the left hand side over here. Interphase. So remember, interphase is when the cell is not dividing, when it's carrying out normal processes. Alright, but at the end of interphase and prophase, beginning of prophase, that is when the cell starts preparing for cell division. For example, the DNA molecules, the chromatin threads, they coil up to form chromosomes, and then we know that DNA replication takes place as well, and then you end up with two DNA molecules. We now call them chromatids, and they are held together by a centromere. So this is a replicated chromosome, and this is just a chromosome, one DNA molecule. So remember, you always count chromosomes by the number of centromeres that are present. So from here, end of interphase, that DNA, the thread-like chromatin, would have coiled up to form separate chromosomes. So, each of those are a very tightly coiled DNA molecule. And then DNA, we can call it replication, occurs so that each chromosome consists of two chromatids joined by a centromere. Okay, and they sometimes draw it like that, but we're actually talking about, one DNA molecule, there's the centromere. It's not actually a physical structure, it's where they are held together. Okay, so what else do we see? Alright, so here was chromatin, now it's chromosomes, then replicated chromosomes. We can see here that there's centrioles, and they actually replicate. So remember, centrioles in animal cells only, the two cylindrical structures at right angles to each other in a patch of cytoplasm and called the centrosome. So the centrioles replicate and they start moving towards opposite poles. So here you can see, there they are. It's actually two little cylindrical bodies, and the spindle fibers start to form between them. You can see here as well that the nuclear membrane is starting to break down. Okay, so they've labeled the nucleus, and here you can see, well, there as well, that it's starting to break down even more. And, it's a membrane, so it's proteins and lipids, and they just dissipate into the cytoplasm so that the chromosomes are able to move. So, if we look here now, they've called this early prophase. All right? Chromosomes are visible, etc., becoming shorter and thicker, and then you can see, prophase, they have replicated. So, different diagrams will say different things. Alright, but as long as you understand the process and why it has to happen. Alright, so they've called this late prophase, showing the centrioles moving and that the chromatids are visible, and there's, two chromatids joined by a centromere, and the spindle fibers are developing. I'm not too worried about early and late, okay? As long as you can recognize and tell me why you think it's that particular stage. Alright, so prophase stands for prepare. Okay, then metaphase. Metaphase means middle of the cell, or the equator, the equatorial plane across the cell. So here the chromosomes line up along the center of the cell in an imaginary line called the equator, and you can see how they are attached by their centromeres to the centrioles, which are at the poles. And in plant cells they don't have centrioles, but they tend to have like a region, it's called the aster. And then the spindle fibers develop from that area, and they don't really know why. So they've lined up along the equator, and then we go to anaphase, and anaphase means apart. So the replicated chromosomes joined by the centromere, they are going to move apart. The chromatids are going to move apart. So the centromere divides longitudinally, just fix that up, and each chromatid, we can now call them daughter chromosomes. So chromatids or daughter chromosomes are pulled to opposite poles by the contracting spindle fibers. And they actually do move in this little V-shaped fashion because they've been pulled along by their middles. Alright, they move towards the poles of the cell. So anaphase is for apart. Chromatids or daughter chromosomes are pulled to opposite ends of the cells called the poles. Alright, then we get to telophase. Telophase, remember, two nuclei are formed. So the chromosomes have collected and a nuclear membrane has reformed around them and the nucleolus is reformed. So now we have two nuclei, and you can see here that even cytokinesis has started. Alright, it can, the cell doesn't stop and start each stage. So cytokinesis is already happening. It's like, the constriction of the cell, or sometimes they talk about the cytoplasm invaginates. It's like taking that ball of press stick and twisting it into two, and you end up with two daughter cells, and they are genetically identical to each other and the original daughter cell. So let's look here. Telophase. The chromatids, or daughter chromosomes, reach the poles, new nuclear membrane starts to form, and the spindle fibers, they would have started breaking down. Alright, then late telophase they've got. Alright, the chromatids are now, well, I've called them chromosomes before this, okay? And then they're going to unwind, so they're going to go backwards now. So they were chromosomes, and then they're going to unwind to form chromatin again, the long threads. And these nuclei are now identical to the original one. But there the molecules have already started unwinding and becoming chromatin. So you end up with the two daughter cells, and they've each got the same number of chromosomes as the original parental cell. And then I did explain on this as well that for plant cells it could be quite a problem because they are surrounded by a cellulose cell wall. So there's the cellulose cell wall. So what happens here? Okay, let's just try and draw the chromosomes, nucleus, etc. Alright, so what happens is cellulose gets deposited along the center, and that cell, we call it a cell plate. That cell plate sort of grows from the middle and it grows outwards towards the edges of the cell. So eventually you get a cell plate growing across the whole cell. So switch this off. These people only phone me when I'm doing mitosis. Okay. So you've got, there's the plant cells going to divide by a cellulose plate forming there. I'm just going to draw it sideways over here. So eventually you end up with two daughter cells like that, and they each have their own cell wall. And okay, there's the nucleus and the chromosomes. Alright, so each have their new cell wall because a cell plate grows across to separate the cell into two. Alright, so I hope this is a clearer copy. It looks like it on the screen here, and I will delete the other one so that you can use this one instead. Okay, now let's just see here.